Cleavage is the rapid mitotic division of zygote to form morula and blastula. Firstly, let me just draw the structure of female reproductive system. Suppose this is our fallopian tube and here is our uterus. This is our fallopian tube and here is the another one. And so we know that the zygote is placed in the ampulla region of the fallopian tube. Let me just draw the zygote here. So here is our zygote. And now we will have to discuss about the formation of morula firstly. So the morula is formed due to the rapid mitotic division inside the zygote. So let me just draw the structure of zygote so that we can easily divide. There is mitotic division will take place in the zygote. So this is our zona pellucida. So far we have already discussed that zona pellucida get dissolves before the fertilization because of the acrosome reaction, but at that time we have already discussed that only a small portion of the zona pellucida get dissolved and through which the entry of a sperm will take place in the vitelline membrane. And so after the entry of a sperm, that is acrosome reaction, this very zona pellucida forms once again. And inside of zona pellucida we have already discussed that there is vitelline membrane, and inside of vitelline membrane here is our zygote, and inside the zygote there is our zygotic nucleus. So first division will take place after 30 hours of fertilization. So from which plane first division will take place? We know that there are two poles in the zygote, that is the animal pole and vegetal pole. The animal pole is the one in where the nucleus is towards the zygote. So this is our animal pole and here is our vegetal pole. So the first division will take place from this very axis. So the first division will take place in the zygote from this very axis. So this is our first division in the zygote, and there is no effect of the division of zygote on the vitelline membrane or zona pellucida. So these form two unequal cells because of the division of zygote. Let me just draw the two unequal blastomeres or cells of zygote. So here is the first bigger one and here is the second smaller one, and here is its nucleus. So now after 10 hours of the first mitotic division, the second mitotic division will take place in the larger blastomers. So that is after 40 hours of fertilization, the second mitotic division will take place, and this time the division will take place from this very axis, that is perpendicular to the first one. And now for a short period of time there are 3-celled stages of embryo, and after that there are 4-celled stages of embryo, because this very smaller blastomer also gets mitotic division. So because of this there are 4-cell stages of embryo. Let me just draw the 4 blastomers of embryo. So these are the 4 blastomers of embryo having their nucleus. So these are the 4 blastomers of embryo, and this is up to our 40 to 45 hours after the fertilization. And on the third day of fertilization, these 4 blastomers forms about 32 celled stage, that is 16 to 32 celled stage, which we call the morula. And at that time where should be this very morula? So let me just draw the female reproductive system once again. So here is our fallopian tube and uterus. Here is our, and we know that there is endometrium folding inside the uterus. So these are our endometrium folding, and on the third day, that is on the last period of third day, that is about 4th day, this very morula comes here, that is in the endometrial folding. So this very morula comes here, and so let me just draw the morula once again. So the morula is 32 celled stage, 16 to 32 celled stage of embryo, and these 32 or 16 cells are like this. that is, these are the blastomers. And because of the unequal division, there are two types of blastomers are formed. One is the smaller one, which is outer side, and these are transparent. So these smaller ones are transparent, and these are known as micromeres, and the other ones which are larger, which is inside, are known as macromeres. Now we will discuss the formation of blastula. So blastula is generally formed because these 32 blastomers of morula divides to form 100 to 200 blastomers, and these blastomers arranged in a manner. So these smaller micromeres arranged like this to form a membranous structure which we called trophoblast, and inside of it these are the large macromeres which are accumulated in one side of this very trophoblast, and because of which there is a space is formed, and this very space is known as blastocele, and this very membranous structure which is formed because of the arrangement of micromeres is called trophoblast, and this very structure, that is a knob like structure which is formed due to accumulation of macromeres in one side of trophoblast, is known as embryonic knob. And because of the accumulation of nutrition in this very blastocele, so nutrients comes from the endometrial glands of uterus, which is absorbed by this very trophoblast cell, that is the micromeres, and these are accumulated in this very blastocele, and because of this this whole structure becomes larger and larger, and on the sixth day it becomes so large that the vitelline membrane and the zona pellucida, which is outside of this, get degenerated, and we know that there are three primary germ layers in the embryo, which is known as ectoderm, endoderm, and mesoderm, and all these three germ layers comes from this very embryonic knob. Then what is the function of this very trophoblast? It do not participate in the formation of embryo directly, but it helps in the formation of embryo because it protects this very embryonic knob, and it also provides nutrition. It absorbs nutrition from the endometrial glands, and it also forms the placenta during fetus development. So this is the function of this very trophoblast, or what we call the micromeres, and this site, the site along which this very embryonic knob is placed, is known as embryonic pole, and the site opposite to it, that is this very site, is known as abembryonic pole. And because of the blastocele formation, this whole structure looks like cyst, and so this is known as blastocyst, or what we call the blastula. So this is the whole process of cleavage. That's all. Thank you.